Welcome, welcome. All right, excellent. Welcome back. I hope you enjoyed your refreshments. We have two amazing speakers coming up. First is James Fraser. Where James? There you are. And James, sticking on the theme of accents, James has an accent that he's going to try to hide from you. And whoever guesses it will win the prize, eh? James, take it away.
Here.
Join me in welcoming James.
It's a real pleasure to be here. I've learned so much. Thank you, Larry, and Larry for the invitation. Thank you, everybody, for the conversations I've been having. I've just been enthralled by what I've been learning here. Today, I'm going to talk really about a new project that we've launched out of our lab and involves many folks that really seeks to address this fundamental question of why do drugs cost so much to make? And see if there's something that we can do in academia that isn't just develop new drugs, but actually attack some of these core issues. And really just to whet your appetite on the long-term direction of the talk, drug development really fails both because of issues with targets, right? Either we've picked the wrong target or the target doesn't modulate the biology or the actual molecule doesn't work well enough on the target. But also a lot of the drug development fails because of issues with anti-targets. These are the other proteins that a drug might encounter as it makes its way through the body to the site of action. And those are issues around delivery and toxicity. And today, what I want to talk to you about is really an academic drug discovery story that's first going to focus on targets, and then how that got us starting to think about these anti-targets and how we could bring the same type of verve and approaches that we bring to targets to anti-targets for the first time. So luckily, Art gave a fantastic primer to interferon biology yesterday. Today, I will just say that when viruses get into our cells, that leads to a huge wave of interferon signaling that activates the transcription of many different genes that make many different proteins. One of those classes of proteins that are upregulated are a couple enzymes in the PARP family. PARP is a type of enzyme that is a writer of a post-translational modification. So what it does is it takes NAD and it attaches it to different parts of target proteins, and then that changes the way the proteins interact with each other, where they localize in the cell. And downstream of that, those proteins then control the antiviral response. Right? And I got really interested at the outset of the pandemic in this sort of part of our innate immune system, in part because our cancer center at UCSF is directed by this fellow here, my now dear friend and collaborator, Alan Ashworth, who was very interested for his whole career in PARP drug development in an entirely different branch of biology, because PARP enzymes are also really important in how DNA gets repaired, especially in contexts like breast cancer, and he was integral in the development of some of the first inhibitors of PARP enzymes for breast cancer. So we have this convergence of biology between cancer and virology here. But what many viruses do is they encode a protein that, rather than writing this modification onto target proteins, they're going to erase it, hopefully to counteract the antiviral response. And that class of enzymes are called macrodomains. So viruses encode macrodomains that erase the marks that are put on by PARP enzymes as part of interferon signaling. And these macrodomains are present in very diverse and distinct types of viruses. Some of the hit list of the pandemic preparedness talk from yesterday are on there, and they contain these macrodomains. And they've been acquired many different times throughout evolution from their animal hosts. And so in SARS-CoV-2, which encodes a macrodomain that I'll call MAC1 for the rest of this talk, what it does is it erases these ADP ribosylation marks that are important for the antiviral response and basically allows the virus to replicate. Now here's the weird thing about why you've probably never heard of this as a drug target before, and why it's one of the relatively understudied proteins in coronaviruses even after 5 years. because it has this role in enabling our immune system, there's no good cellular model in a dish for studying how macrodomains work. Because basically, you need the immune system, all these different cells, to be talking to each other properly to get this effect for the virus to eventually be cleared. So if you make a virus that has only a mutation in the macrodomain that gets rid of its eraser activity, in cell culture, at best, you see maybe a 2 to 4-fold effect in highly ginned up conditions. And for a biologist, 2 to 4-fold is basically noise. But if you take it to an animal model, the exact same virus, an animal model that's completely lethal with the wild-type virus, you now get complete survival with just this macrodomain point mutant. And that's seen across many different animal models. So it's just this one inactivation basically unleashes the power of our immune system to clear the virus. Here's the other trouble with macrodomains. When we started working on them, there were no good assays. We basically couldn't figure out how to measure the enzymatic activity. There were no starting points of chemical matter other than what I'm showing here, which is the product of the reaction, ADP ribose. And our group basically said, that's not a problem. We have a solution for that. And our solution is to turn to a structure technique called fragment-based drug discovery. And the way that works is here I'm showing you a molecule of a very typical size that you would find in a screening deck for a high throughput screen in a pharma or biotech company. And I've colored all the different cycles that make up this molecule in different colors here. Fragment-based drug discovery says, rather than screening these large molecules, which are more likely to be potent, let's just screen these cycles, or a cycle and a half. They're going to be harder to detect in some ways because they're going to be weaker, but they're going to be easier to detect in other ways because they're going to be more promiscuous. And that's because we basically get observations of all the good interactions without any of the bad interactions. So if we imagine the protein is this sort of red blob here, the green cycle that sort of impinges upon the protein means that if we were to screen this whole molecule here, we'd have no observation at all. It wouldn't bind at all, and we'd have no information about the fact that the yellow part and the pink part and the gray part actually make good interactions with the protein. So the hope is if we screen those independently, we might be able to glue them back together in some way into a molecule that actually works. And so the way we detect that is using a technique called X-ray crystallography. And the way we do that is we've basically been building a pipeline for doing that at UCSF that is modeled after a facility that we used several years ago in the United Kingdom that's run by my good friend Frank van Delft. And here what we do is we take a 96-well plate, we pipette the protein into each well, we, at very high concentrations, force it to form crystals. Then we take that tray, we use acoustic liquid handling to move very small amounts of compounds next to each individual crystal that's in the tray. And then we take advantage of robotics to put the crystal in front of the X-ray beam to collect diffraction data that then leads to hundreds or thousands of data sets that we have to analyze computationally to figure out whether the molecule has bound. All right, so now I'm going to just give you a primer in how we analyze that data. So here I'm showing you the structure of a potential fragment molecule. The little red stars there are water molecules, and the rest of the yellow stuff there is the protein. What X-ray crystallography gives you is essentially a probability distribution map of what the contents are inside the crystal. And so in an ideal case, you do this calculation and you get a green blob that looks like this. And you don't have to be an expert structural biologist to see, okay, that yellow fragment would fit really nicely into that green blob right there, right? And the red is sort of the areas where there's a little bit of negative noise. The trouble is these fragments bind very weakly, and they often overlap with molecules like water molecules or parts of the protein that are there in our reference state And so these maps, these probability distribution maps of the difference, look something more like this here on the right, which looks nothing like the fragment because the contribution of that glycerol molecule that's there has not been taken away. And so it's even worse because they're only there part of the time, so maybe it's even weaker. It looks like this. And now you can never figure out that this molecule has actually bound in this place. So we think of this as this challenge of how do we deconvolute all the things that are present in the sample, and ideally what we want to be able to do is separate out the ground state, the reference state, from this ligand bound state and be able to model it at very high confidence, and we want to be able to do that no matter where the molecule binds in the sample. And so here the solution is we use a statistical analysis that basically allows us to subtract away the reference state in a very different way. And so here I'm showing you this blue mesh here. It's almost uninterpretable. This is like the overexposed photograph of electron density maps here. And another molecule that you can see in the center, and it's not even completely enveloped by that blue mesh. What I'm going to do now is just play the movie as we start to subtract away with this statistical technique we call PANDA, and now we can reveal just sort of a beautiful map that uniquely defines the position of the molecule. And so we can do that hundreds or thousands of times to discover all the molecules that bind to the protein and then overlay them in the context of the three-dimensional structure of the protein. And so here I'm rotating around a structure of the protein with all the molecules that we identified from this fragment screen. These are very small, very weak molecules that bind all over the surface of the protein. And this gives us a starting point to think about drug discovery. And basically, the analogy to language here is that small molecules, eventual drugs, are composed of letters, the atoms, words, which are the cycles or fragments that we want to combine into sentences with meaning. All the drugs, all the small molecules that we take are just recombinations of different words. And here, there are people from pharma in the audience. I've highlighted some of the letters and cycles there that immediately speak to us that all of those molecules are kinase inhibitors, for example, in the context of cancer. The trouble is that current methods for searching this potential chemical space are very slow, and this chemical space is huge. So, we're trying to find the needle in the haystack, maybe one molecule out of 5 or 10 million that we might be able to screen. The amount of molecules that we can make now is more like finding a needle anywhere on the surface of Earth. And the amount of molecules that we think we could make just using normal atoms is something, ridiculous, like the number of atoms in the solar system, universe, whatever. It's literally an astronomical number. And this purchasable, synthesizable space, that middle category of molecules that we think we could make on Earth today, when we order it from our friends in Ukraine, a company called Enamine, is approaching the trillions of molecules. And that means huge computational problems. We can't even load the database of all of the structures of all of these molecules onto AWS in one instance. So skipping, a lot of work, what we do is we use computation, especially techniques like active learning, to help navigate this vast chemical space. And we're really leveraging the fact that we can start with these experimentally derived words as starting points. We're not just starting from scratch. We know where these words are relative to each other, and then we can connect them into sentences with the meaning of inhibit this protein. So this is the point in the talk, especially at this meeting, where I get asked this question: couldn't we just use AlphaFold for all of this? Especially because, DeepMind likes to put out blogs that AlphaFold now predicts the structure and interactions of all of life's molecules. And I am an AI bio optimist, but I'm just going to take a little time to just show you where we actually are. So in the prediction of how small molecules interact with proteins, contamination in the ligand training data for these AlphaFold methods is a huge problem. And it means the generalizability is much more limited than just in predicting protein structure. So here I'm showing sort of histograms from the AlphaFold training sets that are used to infer ligand protein interactions. And what I hope you can see is that these are heavily biased by specific proteins with high identity to each other, ligands that look a lot like each other, and pockets that are very, very similar in different contexts. And that means the generalizability for predicting small molecule protein interactions is really lagging behind our ability to predict protein structure alone. And a great example of this is in a blind challenge. So this is the CASH challenge that's organized by the Structural Genomics Consortium in Toronto. And we actually volunteered our assays once we developed them to test the state of the art in a blind challenge where this SGC would purchase molecules predicted by computational chemists, and then we would test them and solve crystal structures of them. And importantly, they already had the data from our initial fragment screen. And so we tested about 2,000 molecules, and the best were very, very weak. And then we even gave them, the teams, the results from those and said, improve upon those predictions. And there was basically no improvement in predictions. So that's, I think, a great blind test of where we are with the state of the art. The other thing is that we have all of these structures of small molecules from this discovery program that I was telling you about. And they are relatively diverse. What I'm showing you there is a histogram of how diverse the small molecules are. And what we find is that AlphaFold 3 does a pretty good job of localizing the molecules to the right area, to the right part of the protein. But most of the predictions don't recapitulate the exact interactions that we really need to be able to motivate medicinal chemists to design new molecules, design and make new molecules. Importantly, though, we did notice this interesting power where the AlphaFold confidence itself is actually predictive of how close the prediction is to the ground truth, to our structures. So the models are actually able to maybe recognize similarity in their training set and be super confident, or at least extrapolate a little bit beyond what's in the training set. However, once we add in sort of decoy molecules, these are molecules we know don't bind or bind super weakly, that predictive power almost completely goes away. So what I'm showing you here is a measure of the affinity going left to right, and then that confidence going up and down. And the blue molecules are the molecules we know actually bind, and the red molecules are the molecules that bind either so weakly or cannot be measured, and basically there's no difference there. And this is because AlphaFold has been trained, everything that is input must be true. And so it can't tell what's right and what's wrong. However, if we use even really simple physics-based scoring, like docking developed by my colleague Brian Schoiket at UCSF, we get a weak correlation, but it's at least has some discriminatory power between hits and non-hits. And so this is sort of contrasting those two approaches, and hopefully you can see a segregation of the blue mostly on the left part here, but no segregation going up and down. And so really these are, I'm preaching this combination of experiment and computation together that I think are really enabling this early stage drug discovery in academia. We can now detect these very low occupancy binding events with structural biology essentially as a binding assay, and that yields many starting points, that yields where all those words are relative to each other. And then we can use new computational methods to be able to figure out how to stitch those words together to make more potent molecules. And then the most important thing is, thanks to these breakthroughs in synthetic chemistry from folks like enamine, we now can actually very cheaply and quickly turn the crank on iterating through rounds of drug discovery. So we can get milligrams of these compounds in about 6 weeks for under $100 per compound, right? And that is a capability that did not exist 10 years ago. And that is just transformational for this early exploration there's some crazy stories about how we continued working with them during the outbreak of the war that I'd be happy to tell you as well. But the big lesson is that for us, of what I'm about to show you, is that the very precise structural overlap of these words relative to each other emerge as this key differentiator for success, and these AlphaFold type methods are really not quite there yet to enable that. So again, skipping a whole bunch of chemistry done by Adam and Priya here to combine all of these things, what we have in the middle here is a very potent lead molecule, about a 20 nanomolar molecule, that combines sort of the best of all of our lessons from fragments and docking and machine learning, but combined together using sort of traditional medicinal chemistry. And we validated the heck out of this in vitro, right, with all the enzyme assays and things like that. But we have no sort of cellular model for how this would actually show efficacy in an antiviral study. So really here, it is time to just take a big leap of faith and move from in vitro test tube work straight into animals. And we don't do that just with the first molecule. What we do really carefully is make sure that the molecule can stick around in the bloodstream and get to the site of action. This is a graph on the left here comparing different routes of administration, and then how long the drug stays at a concentration above the red line there where we think it would have efficacy at the site of action, mostly at the lung, but also in the bloodstream. And so this is, the area of pharmacokinetics. And in this case, in this molecule, we could do that with, about twice a day dosing. And so it was time for the critical experiment. So here we're going head-to-head against the protease inhibitor in a mouse model that is normally completely lethal when left untreated. And you can see the protease inhibitor there has about a 50%, 40% survival. And then when we treat with the macrodomain inhibitor, we get, if anything, a slightly increased survival. This is repeatable across sort of several different animal models as well. And so we think this is a really cool, target for many reasons, because it works by unlocking this innate immune response. And so rather than a host-directed therapy, this is really a host-enabling therapy. And I hope your attention is also drawn to this movie that's playing here on the bottom right here. This is showing, the over 1,000 molecules that we made and determined crystal structures of during this project. So why am I playing this movie when on the previous slide introducing you to that lead molecule, I only showed you like 5 or 6? Well, it's because that really, while doing this project, we learned that the really hard part about drug discovery isn't inhibiting the target. We had molecules that worked in a test tube way, way before we had molecules that worked in animals. And probably most of you, are actually semi-aware of this, because I bet you there's a sizable percentage of people in the room that have taken Paxlovid, right? And one of the major issues with Paxlovid is it's not just a SARS-CoV-2 protease inhibitor. It's actually 2 drugs combined into one. So there's one drug that is inhibiting the protease inhibitor. There's a second drug, ritonavir, that is an inhibitor of a cytochrome P450. And that is an enzyme that would chew up and transform the active ingredient that we actually care about for treating coronavirus, right? And so, Pfizer, when they were developing this, was aware of this liability. This was like a huge issue. They tried to get around it. They've gotten around it now. But the really hard thing is this multi-parameter optimization of not just how your drug is going to work with your target, but also how it's going to work with all the other proteins in the body. And so we think of this as, studying all the friends the drug made along the way. And in our field, we call these the ADMET properties for absorption, distribution, metabolism, excretion, and toxicity. Essentially, these are the proteins that generically interact with small molecules to move them around the body, get them in and out of cells, to help the body deal with potentially toxic chemicals by chewing them up or modifying them to neutralize them. The transcription factors that activate whole programs that tell the cell, hey, there is stuff that probably shouldn't be in our cells right now. Let's make more of those enzymes or make more of those transporters. The good news for us, though, is that this is not an infinite list. The proteins that actually mediate this, which we call the anti-targets, are actually a relatively small number of proteins compared to the whole proteome. And they're things like transporters and enzymes and very specific recurrent tox targets. And these are the proteins we were worried about their interactions with during the development of our macrodomain inhibitor. And so here I'm going to show you like a really boring, colorless slide, because this is an example of sort of the types of assays you run against panels of these proteins during a drug development project to try to minimize their interaction with your drug. And so this is an example from our project of a really, really clean example. But most of the time, these properties are sort of the vexing thing once you've already achieved potency on your target that are really slowing down drug discovery and making it really expensive. So, most of the molecules that are made in early stages of hit to lead really would fail to meet these objectives. There's a huge, even clinical attrition due to ADMET issues, particularly tox, but often also metabolism, and the differences between species play a role in this as well. And when we look across historical drug discovery campaigns, I love this slide from Novartis, you can see that when optimizing all of these properties, although the general trend is for them to improve, there's a lot of jagged edges here. There's a lot of 2 steps forward, 1 step back. And we always compare this to really the carnival game of whack-a-mole. Basically, we're on target. And then we've got a CYP issue. Boom, we knock it down. Now we've got a hERG liability. Boom, we knock that down. Now it's no longer getting into the tissue we want because it's not being transported in the right way. Boom, we fix that. And this happens over and over again in drug discovery. So what we really want to do is have a way of systematically and purposefully exploring these proteins. And surprisingly, that's not something that's ever really been done before. And so about 6 months ago, we got some funding from ARPA-H in a project we call the AVOID-OME that is a huge consortium that I lead that involves a software foundation, the Open Molecular Software Foundation, a biotech company in the Bay Area, Octant, and a bunch of structural biologists and geneticists at UCSF to really tackle this problem. And we call the overall effort Open-ADMET. And then right after we received that funding, the Gates Foundation reached out to us, and we then had some add-on funding from them through a collaboration with Schrödinger. In the last few minutes, what I want to tell you about is just sort of the change in mentality of how we're going about attacking these ADMET anti-targets. Rather than sort of only dealing with these issues as they arise in very, very local regions of chemical space that are already on target, which is what's done in a traditional campaign, where you can't really transfer the lessons between program, and you certainly can't transfer the lessons between companies because you're already so advanced in the molecule, you'd be giving up a whole bunch of IP if you shared it. What we want to do is tackle the chemical space that they recognize up front and sort of treat them like targets, figure out the diversity of molecules that they bind to so that we can build better machine learning models to really inform all aspects of drug discovery. And so the way that works is our friends at Octant are really building the fundamental data sets that in a consistent, balanced way with their high throughput Navigator platform that includes sort of assays and also the chemistry. We've already miniaturized and reduced the cost of several of these assays, several orders of magnitude, which is enabling for us. And then now also working on some of the physical properties of the molecules as well in higher throughput. We then can take that data over to OMSF, the software foundation, which is building models, machine learning models, representations of chemical space, where we can say, actually, we know a lot about how CYPs bind this class of molecules, but we know nothing about it over here or over here to feed in where Octane should be making molecules and doing the assays. Active learning is an important part of that. And then, building sort of baseline predictors of all of these activities that in an open source way that any company can then leverage. We've also been running blind challenges. We just had our first AdMet blind challenge to really spur the community as well to improve upon these baseline models. And then structural biology, my passion, is both seeding new regions of chemical space with unbiased screens like the fragment screening approach that I told you, but also enabling outliers and enabling new anti-targets. So we are both figuring out new regions of chemical space to go into, but then also taking those models, looking at where the outliers are. Why are these models not working right now? Trying to figure out the detailed atomistic interactions of what's going wrong there. And then hopefully aligning them so that better machine learning or free energy perturbations can work. And so what we really want to do is create this very open, common good data set for AdMet and baseline machine learning models that the entire community can use, where in the future, this multi-parameter optimization of both on-target and anti-target properties, right, are able to be tackled upfront with the same high-quality data that is really only usually available very, very late in a campaign. And so with that, I will thank the folks who were part of the huge discovery effort for the MAC1 inhibitor that I led. And then also point out Nevin Krogan is the head of this overall AVID center that was at UCSF. That this was one of 5 pandemic preparedness projects. And then thank the folks in my lab, especially Galen Corey, who determined almost all of the X-ray structures that I showed you today. Michelle, who really was a thought partner during the early stages of the AVOID-OME work and helped write the first drafts of these proposals and get me into an ARPA-H friendly mentality. So wonderful to reconnect with her here. And I'll end on a bit of a sour note, which is that the grant that supported that work was recently terminated. And we have a real problem with where the NIAID and other federal agencies will be prioritizing funding for pandemic preparedness, for antivirals in the future. And so do what you can to help fight the good fight in your local districts as well. I'd be happy to take any questions.
Wow, James, I'm sure we're going to have a ton of questions, so let's dive right into it. Thank you for an amazing talk. We'll start down here, and then Larry, and over here. Oh, and back there.
That was great. I was really intrigued by your modular approach, where you're taking small pieces of the small molecules. And maybe this is a naive question, but I wonder if you could do this to predict protein-protein interactions. Could you, for example, start with dipeptides or tripeptides, and then build from there to predict interfaces that promote protein-protein interactions?
Yeah, so, I imagine, where is it? Our friend over there is going to have a lot of thoughts about that approach. Maybe just provide a little bit of historical perspective. That was actually sort of the dominant thinking in protein structure prediction pre-AlphaFold, was this sort of fragment type approach, fragment assembly type approach. Now, more commonly used to sort of put a fragment and then use a diffusion model around it. So there's sort of a convergence happening there. But there's some really key differences between protein space and chemical space. Protein space is just much more limited, and we have all this evolutionary information, things like that. And so there are just other shortcuts that work better in protein space. I think what you're saying would work, but we've discovered better ways to do it in protein space. And in chemical space, we just don't have that. We especially don't have the ability to infer negative examples from not being present in the evolutionary record. in the same way we can with proteins. And that really changes the way deep learning can work because we don't have balance in the right way. Yeah.
Yes, beautiful. How do you think about induced fit with fragments where the affinity of the fragments is really not sufficient to drive a conformational change that might eventually surface? And then how do you think about linking fragments that are adjacent to each other to achieve sufficient affinity?
If they can put the slide back on, I'll show you, that I have a visual answer to that question that can play maybe in the background. So one of the great things about these new approaches for sort of increasing the sensitivity of fragment analysis, we get the protein conformational changes out of that for free. And so we're seeing a lot more conformational selection, induced fit type things, like what I'm showing here, which is from a crystal system with the molecule soaked into it, happening there. I will say that our mentality for linking, right, does have a fairly sort of static element to it. We are aware of the conformational changes that can happen with each of the individual things. We expect to stabilize those when we link things together. This is sort of a disappointing thing for me because I would say my background is much more in protein dynamics than drug discovery. So I would like to eventually really harness motion a lot more and ideas about conformational entropy as a driver of affinity even more. But right now we sort of take the structural excursions from experiment and leverage them where we can. Yeah, great question.
Well, I had a great question, but I know it was a great question because Naboisha just asked it. So,
Great.
you're up.
Okay, this is such a good, solid science, and unfortunately, it also shows that it takes a lot of work and
Yes.
time to get somewhere. I would like to solicit from you kind of a high-level opinion on about some other projects that are beginning, namely virtual cell, artificial organism. How do you see the interaction between what you guys do, which is I think so, but with the comments that I just made, and these other projects that promise a lot. I think I heard even something like in 10 years we'll cure all disease.
They're moving up the timeline. The CZI, Chan Zuckerberg science, it was originally going to be by the end of their eldest daughter Max's lifetime, but now I'm glad to know it's before my kids are even teenagers. So it's, But I think these are wonderful parallel trends, right? And the way I see sort of the open cell type initiatives, virtual cell type initiatives are much more, we have the parts list. We want to figure out how things work together. And mostly right now, the aspirational goal is how would that respond to largely genetic perturbations in the parts list? I'm also a big fan of genetic medicine. Small molecules, I think, are still going to be around for a really long time. Figuring out the effects of these perturbations on small molecule perturbations on sort of virtual cells is like a long-term goal where we should eventually meet. I would say I probably think it's going to be about 10 years before we're there, but I'm long-term optimistic on those two sort of research trends eventually connecting. Yeah.
So this is embarrassing. So in the middle of your talk, I got lost in an incredible way.
Okay.
Incredible for me.
Oh.
I felt like a dope. And I want to make sure I understood what I think you said.
Yeah.
Or that I misunderstood it. So I thought you said, that when a drug fails because of an off-target binding,
Yeah.
that the things it binds to are known often, and they are a completely tiny subset of the proteome.
Yes.
And that is the avoid-OME we're talking about.
Yeah.
I didn't know that.
I'll maybe add a little bit more nuance, right? So a lot of drugs will fail because of toxicity, because of very related targets, right? So, a dirty kinase inhibitor that hits another related kinase, or a GPCR agonist that is hitting something else. So those are going to be idiosyncratic from program to program. But there is a whole bunch of preclinical and clinical failure that is basically just mediated by these guys here, right?
And not just some random essential protein.
And this is what's crazy. And this list was really curated by my great friend and mentor, Mark Mirko, who's a longtime CTO of Vertex and founding CSO of Relay. And, and when he gives a talk on sort of this stuff, he's just like, it's the same stuff, but we never systematically study it. And every single program he has been involved in his career, and I think he's got like a dozen marketed medicines to his name at this point, right, has had these issues in their development path. And if he has a dozen approved medicines that have that, just imagine how many failed potential medicines he's worked on that have been felled by this type of stuff. Yeah.
Up here, and then we've got in orange.
When I look at that list, James, especially the SIPs,
Yeah.
and I think about all the stuff we heard, 10, 15, 20 years ago, and even more recently about pharmacogenomics, and it's just the usual suspects over and over and over again that seem to be killing these things. So how much was this search for lack of a better word, novel pharmacogenomic targets or limitations or whatever, a distraction to going after what would seem to me stuff that was found out in the binding ground? Let's take some liver cells, grind them up, and see what's metabolizing the drugs era.
Oh, I think it's actually a marvelous convergence. That basically one of the big trends from pharmacogenomics is that so much of the variability in this response is these limited set of players. And what's really hard to think about now is if we're going to when people optimize these properties, right, they're optimizing the eventual dosing and eventual this for a majority population. We know that there are these huge pharmacogenomic effects. I didn't talk about, you can see on there for UCSF, I say genetics and structure, and my great colleague and friend Willow Coyote-Maestas is sort of leading that part of the platform, where we're also looking at the genetic variation in all of these anti-targets and figuring out much more mechanistically, right, how we can take that into account also upfront, right, in the development, so that we can have medicines that work for the most number of people as possible, and we don't run into these huge problems late in the game, actually, not even in development, but like in treatment of patients where the dosing is just completely off because of a pharmacogenomic issue. Yeah.
I'm sort of curious, James. This is amazing and wonderful, especially thinking about programs going forward. But I'm curious about sort of where I don't know if I want to call it end game, but let's call it end game goes with this. So do you think you can do this well enough so that we can just not screen those guys and take them out of the library, all the ones that bind to the scary anti-targets? Or is it always going to be some kind of joint optimization?
I think it's always going to be multi-parametric, because basically you have potency working on your side, right? And so you don't need to completely eliminate CYP metabolism. You just need to have CYP metabolism at such a level that you're still covering your target in the time that it takes for the next dose to arrive. And so you always want to, potency solves a lot. Like if we had a femtomolar inhibitor of the macrodomain, we probably wouldn't have to worry about almost any of these issues. So it's always going to be multi-parametric, but because you don't have all the right signals at all the right time. And I'd say also because our regulatory environment is increasingly moving away from animal models and other aspects, I think this is going to be something where that multi-parametric optimization has to be prioritized sooner, and we need the data sets to be able to power it. Yeah.
Back here, up here.
Yeah, quick question. Great talk. one is just wanted to plus one on the suboptimal dosing thing. I mean, it's a huge problem right now, right? And so it sounds like you're thinking about how to sort of do that. The other question I had, this is a little exploratory. I don't know if it makes sense, but have you sort of looked into trying to apply the methodology into the sort of exposomic space, right? There's a huge amount of black box of like small molecules and exposure and their implications on, I mean, there's a lot of off target kind of like mechanisms. So just like even like laying out a landscape of like those mechanisms and pathophysiology to like autoimmune diseases and stuff like that.
Yeah, we haven't thought too deeply about it other than just this theme of convergence. I mean, it's really the same, nuclear hormone receptors that are being turned on, a lot of the same GPCRs and other things that are mediating these sort of xenobiotic responses, whether in the context of drug treatment or environmental exposure. And sort of NIEHS is probably the major, funder at the federal level of that. We haven't engaged with them much yet for sort of obvious, reasons recently, but we're initially focused much more on how to enable new pharmacology rather than looking sort of backwards or to the environment. But it is an obvious next step. Yeah.
And noticing time, this is going to have to be our last question.
Okay, I'll try to make this quick. Aren't these ADMET properties or molecules? They should be pretty concentration dependent though, right? And if you're varying across patient samples, do you think there are also a lot of other molecules that we should be concerned about that would be increased in, say, a certain subset of populations?
Yeah, so I think what you're asking is like the proteins that are involved in mediating the ADMET properties, it matters how much of them there are to be able to predict in a forward mechanistic way, sort of how they would act on specific small molecules. Absolutely. And that's actually where we're starting with the pharmacogenomic sort of aspect of this project. Is a lot of the pharmacogenomics is not about how the specific CYP mediates an interaction with that small molecule necessarily, but how that specific CYP leads to it being destabilized or massively stabilized to change the overall levels of that CYP in the liver or in whatever tissue. And so that's the first level assay that we're doing, is one of sort of protein abundance and protein stability to be able to drive these sort of more mechanistic, more physiological models which treat different organs as different compartments and things like that. And then in parallel, also tackling the specific interactions of the genetic variation on specific small molecules and the interaction there. Yeah, it's a great question.
Thank you, James.